So how's the term been? Congratulations on your acceptance to medical school. Good morning, Ashi. Good morning, Bianca. Good morning, Irma.
Good morning.
Good morning, Miriam.
Good morning.
Yep. So I just want to introduce myself. My name is Nick. I'm a third-year medical student. I'm just waiting for my placement right now in Boston, Massachusetts. And congratulations on your, what is this, first 2 weeks of medical school, right? How was it?
Hard.
Hard. Okay. All right. We've been there. Don't worry about it. We're going to pass. Okay. So the thing is this. Every lecture, you have lecture objectives, right?
Right.
Okay. Go over that lecture objective for the first exam. As long as you can answer the lecture objectives, you're good. Okay. Second tip. Every week, right? Have one study partner. So for example, make a group chat every week, closed book. You have only the lecture objectives in front of you, right?
Yep.
And then you go over that lecture objectives with each other, okay? The only time that you open the book is if you disagreed with the answer, okay? That's tip number 2. Tip number 3, DLAs are high yield. Please do it. The sequence on Sakai, do it on that sequence, okay? Don't put it on the last minute because it's DLAs are high yield. Before, the lectures are 4 hours, okay? Now it's cut into 2 hours, guys. But you still have to do the DLAs. For resources, I think Firecracker is good for first term, but for the first year of medical school, I recommend USMLE RX because next term you're gonna have a BSCE 1, which tests your knowledge for the first year of medical school. So I think that's a good investment if you get RX. And if you had the first 8 books, start annotating now as you go over your lessons. Okay. What else? So objectives, you have textbooks. I love the biochemistry textbook. I think that's very good, especially the questions there, right? For histology, you have Lippincott histology. Go over that one for your histology slides. Or there's a website online for all the histology. I love Yale histology. Harvard doesn't have that. So Yale has histology website. Go over that website. It's good. For biochemistry concepts, I find Amoeba Sisters on YouTube. Search Amoeba Sisters. They have this wonderful animation of DNA replication, of transcription, of translation. It makes it fun and easier for you to study.
What is it called?
Amoeba Sisters on YouTube. Amoeba Sisters.
Got it. Thank you.
Yep. It makes it fun. You know, I love animation. I love cartoons. And it makes it stick, especially those pesky names of your enzymes and all of the others. What else? So I'm not affiliated with DAS, okay? Not anymore. Or this is officially sanctioned by the school, okay? I'm just doing this because I want to meet all of you. All right, so I want this to be interactive. I'm gonna ask some questions. Please answer them, okay? No pressure, no judgment here. All right, 1 more minute, then we can begin. Any questions? Do you have a white coat ceremony?
Not so far.
Not so far. Okay. So you're gonna be in Grenada next term, hopefully.
Hopefully.
Hopefully, yes. Need to get those pesky, what do they call this one? The immunizations and stuff, right?
Oh, I forgot about that.
Yeah. So is everybody okay? Can we begin?
Mm-hmm.
All right. So, good morning, everyone. Bonjour tout le monde. Buongiorno a tutti. Magandang umaga sa inyong lahat. So today we're going to talk about the first 2 weeks of term 1 of medical school. So first you need to know about the organelles. So I need some volunteer. Pick one organelle and let's talk about it. Okay? Any volunteer? Just pick one.
I can volunteer.
Okay, go ahead.
So good organelle, mitochondria.
Mitochondria, okay.
The purpose of the mitochondria is for what's it called, energy synthesis through ATP synthesis.
Okay, so go ahead.
Okay. It's made, well, it has an inner and outer mitochondrial membrane. The mitochondrial membrane is more for, it's kind of like the plasma membrane where it regulates what comes in and out of the cell. And then the inner membrane is more for transporting, what's it called, hydrogen ions in between the inner membrane space and matrix. And then it has multiple cristae, which are multiple folds within. And what I'm missing one more thing in the structure, but I can't think of it.
Okay, excellent, excellent. Thank you for that. So when you think of mitochondria, think about powerhouse of the cell. Think about N-Star. If you're here in the Northeast, we have the power company called N-Star. So it provides energy via what? ATP. So next term, you're going to learn about the electron transport chain. So this happens in your mitochondria. And in the mitochondria, you have those wonderful inclusions called cristae, right? So mitochondria, the origin of this organelle is very different from the others. Why? How does it differ from the others?
Isn't the origin from prehistoric bacterial times?
Exactly. So the endosymbiotic theory, right? So the endosymbiotic theory says that the mitochondria is a bacteria before. It was engulfed by your primitive eukaryotic cell. In terms of disorders, for mitochondrial disorders, is it inherited by all of the children or just the males or just the females?
Just the females.
Just the females.
So if your mother, right, has a defect in the mitochondria, what will be affected? All of the children or some of the children? For mitochondrial disorder. High yield, all of the children. Why is it that all of the children are affected? What's peculiar about the inheritance of the mitochondria?
You can only get it from your mother. So
exactly. Excellent, excellent, excellent. So if your mother has a defect in her mitochondria, all of the children will have a defect in their mitochondria. So it is maternally inherited. Only the mitochondria is maternally inherited. So can you imagine what it means? We can trace now the ancestry of the human race to a woman, to a single woman via mitochondrial analysis, to the mythical Eve. Now, another high yield point. Mitochondria is very important in what? In apoptosis, because it produces something.
Cytochrome C.
Excellent. Excellent. Don't forget intrinsic pathway and extrinsic pathway. If we have a DNA damage, right, what happens? If we have a DNA damage, intrinsic pathway, P53 is induced. If we have the induction of P53, what happens again? P21 is induced. If P21 is induced, what happens next? CDK complex. Your cyclin and CDK complex, they gonna phosphorylate what? They're gonna phosphorylate your retinoblastoma protein. Is it hypophosphorylation or hyperphosphorylation? In terms of there's a DNA damage.
Wouldn't it be hypophosphorylation?
Hypophosphorylation, excellent. And if it is hypophosphorylation, what happens to the elongation factor 2, the E2F? Will it be released or it will be hugged forever by your retinoblastoma protein?
Release.
Careful, we have a DNA damage here.
Oh, okay. So it's gonna accumulate more. Never mind.
Exactly. So you're gonna hug the E to F. So there is no there's no what do you call this one? So the G1 to the S phase transition of your cell cycle will not gonna happen. So let's do it again. Intrinsic pathway in terms of DNA damage, P51 is increased, P21 is increased. You have increased phosphorylation of your CDK complexes, which hypophosphorylate the retinoblastoma protein, which in turn hugs your elongation 2 factor, meaning there's no progression from your G1 phase to S phase of the cell cycle. High yield, could you please give me examples of some mitochondrial disorders that you already know?
Merck and Lieber.
Exactly. So if in the vignette you have some ragged red fibers, and all of the siblings have that disease. So think about mitochondrial disorders. All right. So I forget about the apoptotic cycle, right? So you have also the BACs, and then the BCL. BACs are pro-apoptotic, and then what they're going to do to the mitochondrial membrane is they're going to increase the permeability of the mitochondrial membrane, which increases the release of cytochrome C. As someone have said, this cascade of cytochrome C will going to increase the calcium into the cell, intracellular calcium of the cell, and then you have the apoptotic cascade. So that's your mitochondria. We're good so far?
Yeah, can I just ask you, are they going to ask specifically like that it's CDK4 and 6 for that?
I don't know, to be honest with you.
Just
but,
know. Okay.
But just know CDK complexes, right? To hypophosphorylate the retinoblastoma protein. Okay. They change the exams every term.
Yeah.
Right. Okay, we're good so far. Do you like this kind of review? It's inclusive of everything. Okay? Okay, next. Another volunteer, please. Pick one and talk about it. Pick one. Who's going to be a volunteer?
I guess I can go. I'll do the rough ER.
Rough endoplasmic reticulum. Why is it called rough?
Because of its appearance, I think.
Yes. What's with the appearance? What's the main function?
The
plasmic reticulum.
The proteins.
So rough endoplasmic reticulum, think about your protein synthesis. Okay, here's a high yield question. After the protein has been synthesized from your rough endoplasmic reticulum, where does it go?
Plasma membrane, lysosomes, and secreted.
Okay, so from the rough endoplasmic reticulum, it will gonna go, don't forget, cis Golgi to the trans Golgi, and then from the Golgi apparatus, whether it can get out of the system via exocytosis, get out of the cell via exocytosis, or be degraded, right? So rough endoplasmic reticulum, think about protein synthesis. Okay, another one. Pick another organelle. Hello?
Okay, I can do one. I'll do the nucleus.
Oh, nucleus, interesting. What about the nucleus?
So it's the largest organelle, and this is where we find our DNA.
Okay.
And it's double membrane. So we have the inner membrane that's in contact with the nuclear lamina to stabilize the nuclear envelope. And then we also have the outer membrane, which is continuous to the rough ER that we just discussed. And it has pores to allow the transport of molecules. This is also where we find the nucleolus to make the ribosomes.
Excellent.
And we have chromatin, which can be like active, like euchromatin, or inactive, like heterochromatin.
Excellent. Very good. Very good. So you have the nucleus. Think about it as the command center of the cell. This is the site of your DNA replication, also your transcription, right? And as she said, you have the nucleolus there. That's the site of your ribosome synthesis or rRNA synthesis. You also have chromatin there. What's the difference between euchromatin and heterochromatin?
Euchromatin is less condensed, which makes it more transcriptionally active and it's going to stain lighter. And then heterochromatin is more condensed, so it's not transcriptionally active and it's going to stain darker.
Exactly. So think euchromatin sounds like true chromatin, so it is expressed, right? Euchromatin sounds like true chromatin, so it's expressed, right? How about heterochromatin? Think about it as highly conserved, so it is not expressed. How are we going to regulate the expression of your chromatin then, of your DNA? How are we going to regulate when you want some expression of your chromatin?
Methylation.
So remember your histones? So your histones, right? So the DNA, they wind up through your histones. How many times?
Twice.
Twice, right? How many histones do you have?
Total of 8 with
Right.
I'm sorry.
Ocho,
Thank you.
right?
Thank you.
So you have 8 chromosomes, right? You have H1, H2, H3, and then H4. I'm sorry, not H1. You have H2, H2B, and then H3, and then you have H4. So you have 8 octamers. So the histones, right? They regulate the expression of your DNA, whether it is expressed or not. What kind of amino acids can you find in the histones?
Lysine and arginine.
Arginine and lysine. Why? Why do you think? Your basic amino acids, your positive amino acids, are rich in your histones. Remember DNA? What is the charge of a DNA?
Negatively charged.
Charge, and you need to have a positive charge in your histones, remember? So they can stick together. We're going to talk about that more when we go over DNA replication. And you have a linker DNA, what do you call that linker? That links your nucleosomes from one another. H1. H1 is your linker DNA. We're going to talk about this more next when we talk about DNA replication. Okay, you also have some nuclear pores there, right? Remember, after the pre-mRNA transcript, we call this one the hnRNA, is removed of its introns. It will going to exit the nucleus. It will going to go into the cytoplasm via your nuclear pores. So you actually have nuclear pores there. Okay, another organelle. We're on a roll here. Give me another one.
Smooth ER.
Smooth ER. Okay. Tell me something about the smooth ER.
The smooth ER has no ribosomes, and they are basically used for lipid metabolism and detoxification.
Excellent, excellent. So when you think about smooth endoplasmic reticulum, they don't have any ribosomes. Think about lipid synthesis and detoxification. Lipid synthesis and detoxification for your smooth endoplasmic reticulum. Another organelle.
Centriole.
Centriole. Okay, tell me something about the centriole. This is very, very important.
They are filled with, I think it's microtubules that attach to the chromosomes when they line up to the metaphase plate. Centrioles migrate to the opposite sides of the cell, and I'm not sure what was it that was it the enzyme telomerase? I'm not sure, but that's about all I know about centrioles.
Okay, that's all right. That's all right. Very good. So let's talk about the microtubule. He's talking about microtubule. So when you think about microtubule, I want you to think dynamic instability. that word, dynamic instability. What do I mean by dynamic instability? Anyone?
Maybe
Dynamic
sometimes
instability.
it's stable, sometimes it's not.
So think about it. Think about it. So it is continuously being made and continuously being undone, right? Why? Remember the microtubule, it has a polarity. It has a positive end and it has a negative end. On the positive end, you have the site of polymerization. You're building up the microtubule. On the negative end, you have the site of depolymerization. You are removing this microtubule. So continuous assembly and disassembly, think about dynamic instability. Now, microtubule also, they're very, very important. There are proteins there, your intracellular transport protein. Can you give me the two most important intracellular proteins in the microtubule?
Is it tubulin? Oh, dang.
Intracellular vesicular transport. Anyone? It starts with a K and D.
Thymine and tennessine.
Excellent. So you have intracellular vesicular transport. You have your kinesin and dynein. Now, high yield, right? Which one does anterograde transport?
Kinesin.
Kinesin. Anterograde, positive, right? Away from the axon. How about retrograde transport?
Dyne.
Dynein, meaning going towards the axon. You're gonna learn more about this, I think, next term or on the second module. So when we have diseases like, for example, rabies, for example, Guillain-Barré syndrome, what is happening is the dynein is affected. So it affects retrograde transport. You have your Negri bodies in your rabies. It will gonna ascend towards the central nervous system. And for example, for the GBS, if you've eaten raw chicken, you can get jejuni infection, and you can have ascending paralysis. Okay, so kinesin and dynein, intracellular vesicular transport. Another thing about the microtubule, it's very important in what? In your mitosis, right? So you have the metaphase plate. It makes them aligned into the metaphase plate. Examples of microtubules. Some examples.
Cilia.
Excellent. Cilia. What else?
Flagella.
Cilia and flagella. So flagella is important. You have a 9+2 arrangement, remember? So you have the axoneme, correct? And high yield point. Give me an example of a disease that has a defect in your axonemal dynein. What is a disease? If I have a defect in my axonemal dynein, it's a microtubule defect. A, myasthenia gravis. B, Cartaginer syndrome. C, homocystinuria. D, achondroplasia. So think about Cartaginer's syndrome. Okay, Cartaginer syndrome, there's a defect in your axonemal dynein. So what happens there? What can you see in a patient? You can have dextrocardia. So the apex of your heart, instead of in the fifth intercostal space, it's on the left side. It is on the right side. So you have dextrocardia. You can also have infertility. Why do you have infertility in Cartaginer syndrome? Think about it. There's a problem with flagella. So can anyone explain to me why do we have infertility in Cartaginer syndrome?
I don't know for sure, but I'm assuming it's because the sperm are unable to get to where they need to go.
Excellent, excellent. Remember your sperm, at the end of your sperm, you have those flagella. So if you don't have the flagella, you have a microtubule defect, then it will not gonna go anywhere, right? So there's a defect in the motility of your sperm. Contrast this one. Contrast the infertility of Cartaginer syndrome with cystic fibrosis. What's the difference? They both have infertility, but different reasons. Could you please tell me the reason why you have infertility in cystic fibrosis?
Isn't that the lining, the structural lining of your lungs, whatever's supposed to create that isn't working anymore?
And there's another one. Is this a qualitative defect or a quantitative defect for cystic fibrosis?
Qualitative?
The quality of the sperm, or there's something?
Oh, no, sorry, quantity, sorry.
So there's something actually wrong with the pathway of the sperm. So the sperm itself is okay. Normal sperm count, normal motility. But for cystic fibrosis, think absence of vas deferens, high yield. When you see in the vignette, absence of vas deferens, it means there's nothing wrong with the sperm itself. Think about cystic fibrosis. Give me examples of drugs that inhibit your microtubules that you've learned. Your microtubule inhibitors.
Anti-cancer drugs?
Exactly. Your cancer drugs. Can you give me an example?
No.
So your taxanes, term 4, you're going to learn about your taxanes, your paclitaxel, your vincristine, and your vinblastine, your microtubule inhibitors. Another one is colchicine. Colchicine is very important. Why?
It prevents polymerization of microtubules.
So it arrests them in metaphase. And this is good if you do what kind of genetic analysis? If you want to arrest them in metaphase, because you want to do a genetic analysis, but what kind of genetic analysis? Is it PCR? Is it karyotyping? Is it
karyotyping?
Excellent, excellent. So it is for karyotyping. You want them nicely arranged in the metaphase plate, right? So that's your microtubule. Okay, let's move on. Give me another organelle. Pick one.
Lysosome?
The lysosomes. Okay, tell me something about the lysosomes.
They are hydrolytic, they're acidic, and breaks down a lot of micromolecule in the body.
Exactly. So when you think about lysosome, think about the garbage trucks, okay? They digest all of those unnecessary things in the cell. By what mechanism, though? How does the cell know that a thing is dangerous to itself and you want to digest that? How does the lysosome know that it's that thing is harmful?
Macrophages?
Think about a molecule that the Golgi attaches to something. A Golgi, if something is wrong with this molecule that's been synthesized, it will gonna attach something to that one, and that signals a lysosome to interact with it and eat it.
Well, the Golgi will put the mannose-6-phosphate onto it so it will get sent to the lysosome.
Excellent. So think about mannose-6-phosphate. Pathology integration. Let's integrate some disease. What is a disease? If you have a defect in your mannose-6-phosphate, you're going to have what?
Is it eye cell disease?
Excellent. Very good. Very good. So you can have eye cell disease. So if there's a defect in mannose-6-phosphate, think eye cell disease. Or there's a defect in an enzyme, N-acetylglucosamine. Now, give me clinical presentation of someone who has eye cell disease. Any volunteer? So clinical presentation of eye cell disease, what are we thinking?
They have issues with their nervous system.
Okay, so there's something about abnormal facies, right? And when you look into the microscope, what happens? You can see cytoplasmic inclusions there. Remember, the waste, it's going to build up on the cell. So that's for your lysosomes. Think about the garbage truck, right? Eats all of these harmful elements inside the cell. Think mannose 6-phosphate deficiency. Think eye cell disease. Okay, pick another one. Any volunteers, please?
I was gonna ask you, what is left?
Hello?
I can do peroxisome.
Okay, peroxisomes. Can you tell me something about the peroxisomes?
So peroxisomes are known for very long chain fatty acids.
Excellent.
And I think the disorder associated with that is Zellweger syndrome, where the peroxisomes aren't functioning properly. What else?
Very good.
Yeah, and they're also important in detoxification in liver cells as well.
Excellent. So you have catalase in there, right? So let's summarize. When you have the peroxisomes, think about you have catalase. And why is it important for catalase? Because for alcohol, right? To degrade alcohol, you need to have the catalase enzyme. And also think about beta oxidation of your very long chain fatty acids. You're going to learn next term about the exact pathway of beta oxidation. And she talked about Zellweger syndrome, Zellweger disease, which is abiogenesis. So you don't have peroxisomes. Next term, you're going to learn about Refsum disease, which is there's a deficiency in your beta oxidation. So all of the fatty acids cannot undergo beta oxidation. You're going to undergo omega oxidation. And this omega oxidation increases phthalic acid into your body. So peroxisomes, think about catalase, think about detoxification of alcohol, think about beta oxidation of your very long chain fatty acids, think about Zellweger syndrome, think about Refsum disease. Okay? Pick another one. We're almost there. Pick another organelle, please.
Plasma membrane.
Okay.
So it's a phospholipid bilayer. It has cholesterol in the inner leaflet and the outer leaflet. In the inner leaflet, we also find phosphatidylserine that moves to the outer leaflet during apoptosis.
Excellent. That's a good point.
Polar and charged molecules can't enter into the membrane due to it being hydrophilic, and lipid-soluble molecules have an easier time, or small non-polar molecules.
Good job. So think about the phospholipid membrane. So you have the phospholipid head, the polar head oriented towards outside of the cell, right? And then the inside, you have those lipids. So the polar head is hydrophilic, loves water, but the fatty acid tail, it's hydrophobic, right? So as he said, lipids, they can just go in because you have that inner lipid loving layer, right? However, polar molecules, they cannot go in. So how do proteins or how does enzymes go into the cell? For example, if you have insulin or glucagon, all of these hormones, they cannot just go inside the cell, right?
Transporters.
Excellent. So you can have transporters, right? Water, can it just go into the cell?
They can, but they also use aquaporins as well.
Yeah. Yes. That's a good point. Yes, but they can go just into the cell via what process? Simple diffusion, right? Simple diffusion. You can just go in. How about if I have insulin, or for example, glucagon? How am I going to get inside the cell?
Wouldn't you need facilitated diffusion?
Facilitated diffusion, of course, that's what you need, some protein transporters, right? Did you talk about tyrosine kinase receptors or not yet?
It was mentioned, but not really fully discussed it.
Okay, so when you think about tyrosine kinase and non-tyrosine kinase, what's the difference? Tyrosine kinase have intrinsic phosphorylation of your kinase. Examples that uses, example of hormones that uses tyrosine kinase receptors, insulin. That's very, very high yield. And then you have the MAP kinase pathway. Contrast this one with the non-tyrosine kinase receptors, which doesn't do autophosphorylation, but instead phosphorylates JAK-STAT. So think tyrosine kinase, think about insulin, think about MAP, right? MAP. When you talk about non-tyrosine kinase, think about cytokines, think about TNF alpha. Think about interleukins. You're gonna learn more about this in term 3. And think there is no autophosphorylation. How does it enter then? How does it affect transcription of the DNA? It does via JAK-STAT pathway. Okay, there's another one that I wanted to talk about, a transmembrane protein. It's 7. This is a transmembrane protein that spans the phospholipid membrane. So you have increased cyclic AMP, increase in protein kinase A. What do we call this receptor?
Integral proteins.
So did you talk about G-coupled protein receptors? GS, GI, and GQ?
Not yet.
That's next week.
Not okay. That's
Yeah,
for next week.
next
Okay, I'm getting ahead of myself. So we're good. So we're good so far, right? I think we talked about everything. What else we didn't talk about? Oh, intermediate filaments. Intermediate filaments, they're not that high yield, but when you go to term 4, this is very important in cyto. So immunochemistry, right? You need to stain this one. You need to stain the intermediate filaments to know if you have cancer. So examples of your intermediate filaments, your vimentin, your desmin, vimentin, if you have mesenchymal cancer, your desmin, if you have a muscle cancer, right? Intermediate filaments. How about Golgi apparatus? Could you tell me something about the Golgi apparatus?
I'll go ahead.
Yes, please.
So you have the Golgi apparatus, and then you have the area that is directly in contact, not directly in contact, but it's the one that transports things from and to the rough endoplasmic reticulum. And that would
Excellent.
be the cis Golgi, and then the trans Golgi faces towards the just the cytoplasm to export proteins or just molecules toward, like, in the cytoplasm. And you can do O-glycosylation, which is a post-translational modification of proteins, and it's mainly towards the amino acids that have hydroxyls, which would be serine and threonine. And I don't know. It's the UPS of the cell. It sends stuff.
Exactly. Excellent. Molto bene. So you have the Golgi apparatus. Think about the Amazon distributing center, right? So all of the proteins from the rough endoplasmic reticulum, it will gonna go to the cis Golgi. Then it will gonna be packaged. It will gonna go to the trans Golgi, and then either through the cytoplasm or outside via exocytosis. Now, there are two transport that's very, very important. High yield, your COP1 and your COP2. All right. So think about 2 steps backwards and 1 step forward. COP2 for retrograde transport. If you want that product to be sent back to the rough endoplasmic reticulum, you need to use COP2, COP1 for anterograde transport. If you want the product to move from the RER to the cis Golgi and then from the cis Golgi to the trans, you need COP1. So 2 steps backwards, 1 step forwards. How about microfilaments? What's the function of the microfilament? Hello?
Microfilaments are made up of actin, and they're used like in microvilli and stereocilia, and also for locomotion of the cell.
Excellent. So, very good. Très bien. So, when you think about microfilament, think about locomotion. Think about diapedesis. So, it is for actin, and you're gonna learn next module about the actin and myosin cycle, about the power stroke, about muscle contraction. Right? That's your microfilament. I think we're good so far. Thank you for your participation. Let's summarize them. Okay, let's go to the next slide. So you have the membrane structure. It's a lipid bilayer, as I've said, hydrophilic head, the face is exterior, and the cytosol in the fatty acid tail, it creates a hydrophobic core. Don't forget exocytosis, right? COP2, and then you have COP1. So it is regulated, right? You have a stimulus, and then you have calcium influx. The calcium influx is the reason why you have a fusion of your secretory vesicles with your plasma membrane. Constitutive just means the substance for export is continuously delivered to the plasma membrane. So first you need to have a ligand, it binds to the receptor, and then you have calcium influx. When you have calcium influx, then you have the fusion of your secretory vesicles with the plasma membrane. This is exocytosis. How about endocytosis? When you think about endocytosis, think about clathrin. Okay, clathrin. It is clathrin dependent. So the clathrin molecules, they interact with your receptors and they form a basket-like cage. Look at this here. So this basket-like cage, it drives vesicle formation. You also have dynamin there mediating the vesicles pinching off. But when you think endocytosis, think about clathrin. Clathrin endocytosis, specifically receptor-mediated endocytosis. Oops. When you talk about pinocytosis is cell drinking. This is clathrin independent. Pinocytosis, cell drinking, clathrin independent, and phagocytosis is via your large particles. Endoplasmic reticulum, as I've said, 2 regions, smooth ER and rough ER. Smooth ER, lipid synthesis, detoxification. Rough ER, protein synthesis. Golgi, COP1, retrograde transport, COP2, anterograde transport vesicles, and you have lysosomes there. Remember, mannose-6-phosphate and the Golgi apparatus to target your proteins to the lysosomes. That's how the lysosomes recognize the things that needs to be eaten, right? Autophagy. Inclusions, this is also high yield. Remember lipofuscin. Lipofuscin is a brownish gold pigment. This is wear and tear pigment, right? It accumulates over the years for your lipofuscin. Hemosiderin, this is iron, right? If you have hemochromatosis, you can see hemosiderin. Melanin, of course, brown pigment, now our skin. But for the inclusions, don't forget lipofuscin. That's high yield. Is it normal to have lipofuscin? Yes, it's wear and tear pigment. Peroxisomes. When you hear the word peroxisomes, think about catalase. Why is this important? It is important in detoxification of ingested alcohol. It's also important for beta oxidation of enzymes, fatty acid oxidation, right? If we have a disease in peroxisomes, for example, abiogenesis, you don't have any peroxisomes, you have Zellweger syndrome, right? Or cephalocaudal-renal disease for your Zellweger syndrome. Don't forget also Refsum disease. Microtubule polymerization, positive end and negative end. Think about dynamic instability. Don't forget GTP dependent for your microtubules. And you also have high yield tau proteins. Tau proteins is important in the pathophysiology of your Alzheimer's disease, because in Alzheimer's you have hyperphosphorylation of the tau, which causes neuritic plaques in your brain. That's why you have dementia in Alzheimer's. So microtubules, dynamic instability, positive end, negative end, GTP dependent. You can also have some associated proteins, for example, the tau proteins, which is important in the pathophysiology of Alzheimer's disease. Mitotic spindle poisons, don't forget colchicine, an anti-cancer compound. This is this is administered to a cell to arrest them in metaphase for karyotyping, right? Also, you have vinca alkaloids, your vinblastine, your vincristine, Taxol, an anti-cancer drug. What's the difference between your vinca alkaloids and your Taxols? What do you think? So your vinblastine, vincristine, it prevents assembly. It prevents assembly of your microtubule. However, the Taxol, it prevents disassembly, right, of your microtubule. So different mechanism of action. Vinca alkaloids, it prevents them to assemble, and Taxols, it prevents them to disassemble, right? So it stabilizes and prevents microtubule disassembly. Phalloidin, this is also high yield. Don't forget that it is toxin found in what? Mushrooms. So your Amanita phalloides, it is also used in cytoskeleton research. So they are responsible for toxic effects in the liver. You can have liver and kidney failure and death 48 days after consumption. Why? They bind to your actin, your microfilaments. So that's high yield. When you think Amanita phalloides, think about angel cap mushroom, think about disruption in the function of actin. So you cannot have cell movement. As I've said, microtubule motors, dynein and kinesin, dynein going to the negative end, kinesin going to the positive end. Cilia, it's a microtubule, an example of a microtubule. It's motile, high yield, 9+2 microtubule arrangement. You have dynein arms that has motor protein and it binds your adjacent microtubule. What's the function of a cilia? It moves fluid and particles along epithelial surfaces. It is anchored to the cell via what? The basal body, high yield. So the basal body is used by the cilia to be anchored to the cell. How do I know basal body? These are thin dark staining band at the base of your cilia. Look at the image of the cilia here. So you have the 9+2 arrangement, right? Microtubule arrangement. You have 9 pairs here, 1, 2, 3, 4, 5, 6, 7, 8, 10, and you have your central axoneme, right? Make sure to know that image. That's a very high yield image. In terms of cell movement, you need actin. You need microfilaments, right? So examples of this: filopodia, lamellipodia, pseudopodia. Just know the definition of each. Let's talk about cell death, necrosis, and apoptosis. So let's do this again. Apoptosis, right? Intrinsic and extrinsic pathway. Intrinsic pathway, if we have DNA and cell damage, what happens?
Cytochrome C released from the mitochondria.
Okay, let's do this systematically, right? Let's just review it. So if I have DNA damage, what happens to P53?
P53 arrests the cell proliferate like the cell cycle, and then it also really goes to another genetic proteins to trying to fix the problem with the DNA.
Okay, very good. So just for clarification, because I think you're gonna learn more of this in term 4. But for now, just remember, when you have DNA damage, P53 increase, then P21, then increase CDK complexes, then hypophosphorylation of retinoblastoma. It hugs the elongation 2 factor. Then you have the BAX. Remember, you have 2 proteins, BAX and BCL. How do I know that BAX is pro-apoptotic? There's A in the name. BAX, B-A-X. There's A there. So if there is A, it should be pro-apoptosis. This BAX will gonna go to the mitochondrial membrane. What happens? It increases the permeability of the mitochondrial membrane, which induces the release of cytochrome C from the mitochondria. This cytochrome C will gonna induce your cascade. You have increased intracellular calcium, it will gonna induce your apoptotic cascade. Now give me high yield for extrinsic. For extrinsic, what's the ligand for your extrinsic pathway of apoptosis?
Faz.
Come again?
Is it Faz?
Fast ligand, exactly. And term 3, you're going to learn more about your cytokines, specifically your TNF alpha. Okay, that's your apoptosis. So how am I going to differentiate apoptosis from necrosis? I know this is not politically correct, but apoptosis, think about suicide. Only one cell is affected, right? Think necrosis, think about mass number of people that's going to be affected. Think about the pandemic, right? Necrosis, think about the pandemic. So apoptosis, it is physiologic, always physiologic. It is normal to have apoptosis. It is programmed cell death. Your cells are going to shrink. Your DNA is going to aggregate. However, apoptosis, it maintains membrane integrity. There is no inflammation that follows. No redness, no swelling. There is no inflammation that follows, no loss of function, right? No surrounding tissue damage. Know the characteristics of apoptosis. Contrast this one with necrosis. There are different kinds of necrosis. When you go to term 4, you'll learn about coagulative, you'll learn about liquefactive, you'll learn about fat necrosis. But for now, necrosis is always pathological. There's something wrong with the body. It is always pathological. You have an acute cell injury. You can have cell swelling, right? But in necrosis, you have a loss of plasma membrane integrity. All of the contents of the cell, they're going to leak out and infect other cells. The cell contents are going to be released. You have surrounding tissue damage. You have inflammation. You have redness, rubor. You have swelling, tumor. You have pain, dolor. You have loss of function, lyasa, and you also have fever, calor, right? Those are the cardinal signs of your inflammation: rubor, calor, tumor, dolor, lyasa, right? So please, this is a high yield table. Know the differences between your apoptosis and necrosis. Apoptosis, always physiological, programmed cell death. You have a maintenance of your plasma membrane integrity, no tissue damage, no inflammation. Necrosis, always pathological. There's something wrong with the cell itself. You have cell swelling. You have loss of plasma membrane integrity. That's why cell contents are released and it goes to other cells. So you have surrounding tissue damage, and it is always followed by inflammation. High yield table again. Know the features of necrosis and apoptosis. So let's talk about the cell cycle. So the cell cycle, you have G0, G1, S, G2, and M. High yield. Give me an example of cells that is permanently in G0.
Neurons.
Neurons, excellent.
Cardiac muscle.
Excellent. So your cardiac myocytes, they are permanently in G0. What do I mean if a cell is permanently in G0? What does it mean?
It's not growing.
Exactly. So when there is injury, it cannot grow. So for example, if you have an MI, if you have a myocardial infarction in that part of the heart, the cells are going to die and then it will not regrow. Or for example, in terms of a stroke, if you have cerebral ischemia, right, in the brain, if you have a stroke, the cells die and they cannot regrow. How about the G1, cells that are in G1? Can you give me some examples of cells in G1?
Hepatocytes.
Hepatocytes, okay, what else?
Skin cells.
So think about those cells that are replicating, right? Continuously replicating. So you have epithelial cells on your skin, the lining of your stomach, the lining of your respiratory system. Those are your GI cells. Now, G1 to S. You have P53 and retinoblastoma. They regulate the G1 to S transition. S phase, when you think about S phase, think DNA synthesis, right? This is the place where you have DNA synthesis. How about G2? G2, think about continuous growth of the cell. The cell is continuously growing. And then M, you have your mitosis, okay? G1, S, G2, and M. P53, retinoblastoma, where can I find that? Transition from G1 to S. If there is no injury to the cell can go from G1 to S. If there is injury, it will gonna be stopped by your P53 and your retinoblastoma protein. I have a question, guys. What if there's something wrong with the S phase? I synthesize a DNA that's defective, and then it goes to G2. Do we still have something in G2 that can arrest that mistake? In G2, do we still have that a mechanism there that can remove the mistake?
Possibly the checkpoint in G2 would probably remove it.
Okay, so G2, think about mismatch repair, right? Your MMR, that's very, very important in G2, your mismatch repair. So the mismatch repair, it finds proofreading errors in your G2. You already know a disease that has a defect in mismatch repair. What is that? Clue, it starts with H. Lynch syndrome, HNPCC. You have a defect in your mismatch repair, specifically microsatellite instability. So the microsatellites are just there to provide stability to your DNA. And did you talk about HNPCC or Lynch syndrome or not yet?
We did, but not like the defect of the enzyme or anything like that.
Okay,
Yeah.
so let's summarize the cell cycle, okay? High yield. M phase, cell division. Think mitosis, sister chromatid split, there is no gene expression. G0, think resting phase, senescence. Think about your neurons permanently in G0. Think about cardiac myocytes. G1, growth. Think about growth. Here you have fixed damage bases. Examples of cells in G1, intestinal, epithelial cells, the lining of your GI, the lining of your GU. There is a regulator from G1 to S, P53, the guardian of the cell, and retinoblastoma protein. Examples of drugs that inhibits G1, your tinibs, your imatinib, your tyrosine kinase inhibitors. They stop the cell in G1. When you think about the S phase, think about DNA replication. This is your DNA synthesis. There is proofreading during this synthesis. And then for G2, think about growth. Growth. think about mismatch repair, think about microsatellites. Okay, don't forget HNPCC and your Lynch syndrome. Voila, this is just a summary, high yield summary. You can read that. This is the retinoblastoma protein, right? Retinoblastoma protein. Hypophosphorylation. If I have DNA damage, I hypophosphorylate this one, and it binds my E2F, your elongation 2 factor. That's why you don't have cell progression from G1 to S. Okay. Hypophosphorylation of the retinoblastoma protein leads to no transition from G1 to S. Another G2, another summary here. Okay, another summary. Your normal cells and your cancer cells. Why do we have cancer cells? What's wrong with cancer cells? They don't have p53, right? So you have continuous cell division with damaged DNA. You have continued mutation, selection, and tumor progression. You have increased genetic instability for your cancer cells. For now, cancer cells, you have, think, lack of p53. That's why you don't arrest in the G1 to the S transition. Okay? Proto-oncogenes versus oncogenes. What's the difference? Proto-oncogenes are normal. They are normal regulatory molecules. However, if they are mutated, your proto-oncogenes can become oncogenes. You might ask me, Nick, do I need to know all of the proto-oncogenes and oncogenes? An example of them, yes. So look here on the right image, some growth factors, right? Some transcription factors, your MYC and your FOS, some DNA repair proteins or ATM. You need to know some of them. Errors in cell division. So it's not always perfect when we divide a cell. You can have aneuploidy, and think abnormal number of chromosomes. You can have trisomies, gain of extra chromosome. Example, trisomy 21. Which is your Down syndrome. You can also have some monosomy, mono, one, loss of one chromosome, or nullisomy, loss of both chromosomes. You can also have polyploidy, a whole set of extra chromosomes. You can have triploids and tetraploids. Sometimes the chromosomes are damaged or fragmented. Deletion meaning a chromosome fragment is lost. Translocation, a chromosome fragment attaches to another chromosome. Example, your CML, your Philadelphia chromosome, translocation 922. You can also have inversion. The chromosome fragment reattaches to the same chromosome, but the orientation is reversed. And sometimes you have chromosome duplication, wherein the chromosome fragment is treated as a separate chromosomes. Know the errors in cell division, specifically this one, the trisomies. High yield and some forms of chromosomal damage or fragmentation. You have deletion, translocation, inversion, and chromosome duplication. Now we've moved past cytology. Let's go over tissue histology, right? So epithelium, it's a vascular tissue. It lines exterior body surfaces, and it lines internal closed cavities, right? And some body tubes. It forms glands and ducts. 3 characteristics: high yield, put a star here. They adhere to one another by cell junctions. They exhibit polarity, and they attach to underlying basement membrane. When you think of cancer cells, think about the 3 principal characteristics. Think about loss of these principal characteristics. So in terms of cancer cells, you don't have cell junctions. The cells don't exhibit polarity, and the cells invade the basement membrane. Okay, that's high yield. Classification of epithelium based on number of cell layers and the shape. Just read it, simple, stratified, and special categories. By layers, it can be simple. More than 2 layers, stratified. By shape, it can either be squamous, cuboidal, columnar. Okay. Another example here. Now we go to high yield stuff. So junctional features, right? Zonula occludens. So for this slide, please know the name, the major link proteins that are involved, the ligands. that are involved. Okay? So zonula occludens, tight junction. What protein are we thinking for zonula occludens? What protein is associated with your tight junctions?
Occludin.
Occludins, exactly, and claudins. That's the major link protein for your zonula occludens. What's the cytoskeletal component of your occludins and claudins? Actin. Don't forget actin filaments. Why do we need to have a tight junction?
Not allow paracellular movement of fluid.
Exactly. Very good. It controls the passage of molecules between the cell. So it regulates permeability of the cell. It also defines the apical domain of your plasma membrane. And don't forget, they're also involved in cell signaling. When I say zonula occludens, you say occludins, claudins, actin filaments, permeability, apical domain, cell signaling. Okay? Next, zonula adherens. What major link proteins are associated with zonula adherens?
Adherence.
Cadherin. So think about E-cadherin. What cytoskeletal component is made up of your E-cadherin?
Actin.
Actin. Excellent. So actin filaments. What's the function of your zonula adherens? Anchoring junction cell to cell. So it couples the actin cytoskeleton to the plasma membrane at regions of cell-to-cell adhesion. When I say zonula adherens, you say E-cadherin, actin filaments, and cell-to-cell. It couples the actin filaments to the plasma membrane at cell-to-cell adhesion. Contrast this one with your macula adherens, your desmosomes. When I say macula adherens, you say cadherens, you say desmoglines and desmocollins, you say intermediate filaments, right? And this is also for cell-to-cell adhesion. Focal adhesion, integrins, actin filaments, right? This is also for anchoring to the extracellular matrix. High-yield hemidesmosomes, hemidesmosomes. When I say hemidesmosomes, you say integrins. That's the major protein that's involved there. You also think about intermediate filaments, right? This is important, hemidesmosomes. Why? It anchors the intermediate filaments to the extracellular matrix. Next module, or the anatomy module, you're going to learn about your bullous pemphigoid and pemphigus vulgaris. Did you talk about this in class or not yet?
Yeah, they mentioned it briefly.
So hemidesmosomes, think about bullous pemphigoid, right? Bullous pemphigoid, think about below the basement, below bullous, think about hemidesmosomes. When you think about pemphigus vulgaris, think about your macula adherens, your desmosomes, right? Pemphigus vulgaris, desmosomes, bullous pemphigoid, your hemidesmosomes. How about gap junction? Why is this important? Gap junction. Where can we find gap junctions?
Communication between cells.
Where can we find gap junctions? Give me an organ that has gap junctions.
The heart.
Again, please.
The heart.
Excellent. The heart. Why do we need to have gap junctions in the heart?
Because we have action potential.
Excellent. Go ahead.
Action potentials and like the pacemaker and all of that happening in the heart. So
excellent. So you need the rhythmicity of the heart to be exact, right? You don't want the atria to be contracting first, then the ventricle a couple of minutes later, right? So you have continuous rhythm and rate of your heart. When I say gap junctions, you say connexin, and it creates, what's the function? It creates a conduit between two adjacent cells. It can be found in the heart. So summary, know the classification, know the Latin names, know the major link proteins, know the ligands, extracellular ligands, and the cytoskeletal components and the function. It's summarized here on the slide. Do I need to know this? The tubular secretory and alveolar secretory. Just know the names and some examples of them. I think simple duct, compound duct. Simple duct, the duct does not branch. It can be simple tubular or simple branch tubular, simple alveolar, simple branch alveolar. If it's a compound duct, you have branching of the ducts. It can be compound tubular, multiple tubules, or alveolar, which can be found in your mammary glands. Know the names, know the definition, and examples. Okay, let's go to biochemistry. You're still feeling good? Are you all okay?
Yeah.
You want to take a break, or you're good to go?
I'm personally good to go.
We can continue.
Okay, let's roll then. So DNA replication. What's the difference between DNA and RNA?
Double-stranded versus single-stranded.
Excellent. Double-stranded versus single-stranded. What can we find in the 2' carbon of RNA that is not found in DNA?
Hydroxyl.
Exactly. So you have hydroxyl group in your RNA, right? Which one has more errors in them? Prone to errors?
RNA.
RNA. Why do we have more errors in RNA?
There's no like checking mechanism for RNA.
Excellent. Very good. So you don't have 3' to 5' exonuclease activity in checking damage to your RNA. So DNA, as you've said, deoxyribonucleic acid. What's the difference between nucleotides and nucleosides?
Nucleosides don't have a phosphate.
Excellent. So nucleosides, they don't have a phosphate. So give me examples of nucleotides that you know. Some nucleotides.
Guanine, thymine, uracil. Cytosine.
Excellent. So you have adenine, thymine, cytosine, and guanine. What's the pairing? So they pair with each other, right? Because no man is an island. It needs to have a pair. So adenine pairs with what?
Thymine.
Thymine, correct. AT. How about cytosine?
Guanine.
Guanine, right? CG. Uracil is found only in what? DNA or RNA?
RNA.
RNA. Now, ATCG. Which one? They are bound to each other, but by what kind of bond?
Hydrogen bonds.
Excellent. Hydrogen bond. Which one has more hydrogen bonds?
GC.
GC.
GC, right? So think about crazy glue. CG, think about crazy glue. So they are glued to each other, right? 3 hydrogen bonds. What's the significance of that CG bond? So what if it has 3 hydrogen bonds?
More difficult to separate.
Exactly. So you need more melting energy, right? Increase in melting temperature in order for that bond to separate. In terms of DNA replication, where do you think it starts? Where is the origin of replication? Is it more CG bond or more AT bond?
More
AT bond.
Because you're able to
in terms of DNA replication, it starts in AT bond. Why? It's much more easier to break. You only have 2 hydrogen bonds there, right? And we talked about the DNA. How is it looped around the histones? We're talking about arginine and lysine, positive, and we talk about DNA as negative. We talk about H1 that linkers one nucleosome to another nucleosome. We talked about euchromatin, true chromatin, fully expressed. And then we talked about heterochromatin, highly conserved, which is not expressed. Question, DNA methylation. What happens if I methylate the DNA?
It's not
heterochromatin.
Excellent. So DNA methylation makes the DNA mute. Okay, DNA methylation makes the DNA mute. So you have more heterochromatin. You have no expression of DNA. How about histone acetylation? If I acetylate the histone itself, what happens?
It makes euchromatin, so it's more active.
Excellent. So histone acetylation makes the DNA active. Okay, so the mnemonic is acetylation makes the DNA active. Methylation makes the DNA mute, right? So don't forget that. Okay, now let me see. DNA, AT and CG bond, nucleoside and nucleotide. So let's go about DNA replication. So you need to unzip or unwind the DNA, right? So what is an enzyme that unwinds the DNA?
Helicase.
Helicase. Question. Let's have, what if I have a deficiency in helicase? What do you call that disease that's associated with helicase deficiency? So think about Bloom syndrome. B-L-O-O-M, Bloom syndrome, right? Orlando Bloom, think about him. Bloom syndrome, what is a defect? BLM gene, okay? So there's a defect in your BLM gene. Think about Bloom syndrome, deficiency in helicase. So we have the helicase, it goes there, it unwinds the DNA, right? And then the DNA is read in what direction?
5 to 3, no, 3 to 5.
3' to 5'. And the DNA is being synthesized in what direction?
5' to 3'.
5' to 3'. Excellent. Can DNA replication just occur, or it needs something in order to begin?
It's a primer.
Excellent. So you need to have a primer there. So you have DNA polymerase 3, right? So you have the primer there. Which one is continuous production of DNA? Is it on the leading strand or on the lagging strand?
Leading.
The leading strand, it is continuous, right? And the lagging strand, you don't have. Since you don't have a continuous replication in the lagging strand, what do we call those fragments?
Okazaki fragments.
Okazaki fragments. Now, the Okazaki fragments can be linked together by what enzyme?
Ligase.
Ligase. So you need to have ligase. Now, you need to prevent the leading strand from annealing to the lagging strand after the helicase had unwind the DNA. What proteins help with stabilization of your leading strand and lagging strand so that they don't anneal to each other?
Single-stranded
binding. Excellent. Single-stranded binding proteins. As you unwind the DNA, you have supercoils. How am I going to relieve the supercoils?
Topoisomerases.
Topoisomerases. There are two kinds of topoisomerases. What's the difference between topoisomerase 1 versus topoisomerase 2?
1 is more for DNA. 2 is DNA gyrase, which is more for RNA or bacteria, excuse me.
So by the name itself, topoisomerase 1, it only relieves supercoil by nicking at one strand. That's why it's called topoisomerase 1. Topoisomerase 2, it nicks both strands to relieve supercoil by introducing a negative supercoil. You're going to learn more of these probably next module. So can you give me a class of drugs that's topoisomerase 1, that affects your topoisomerase 1? Your irinotecan, your topotecans, your tecans, or your top 1. How about topoisomerase 2?
Etoposide and teniposide.
Excellent. Your etoposide, your tenoposide, right? These are for your cancer, anti-neoplastic medications. We're going to learn more in term 4. And someone said DNA gyrase. Give me a drug that inhibits a subtype of topoisomerase 2 called DNA gyrase.
Is it cyclohexamide?
Excellent. Ciprofloxacin, your fluoroquinolones, right? Your fluoroquinolones, they inhibit your DNA gyrase. You're going to learn more in the mechanism of action and all of the side effects in term 4. Okay? So what's the role of the primase? I forgot about this. The role of the primase is to synthesize what by the name itself?
A primer.
A primer. So you have the primase to synthesize the primer. Does the DNA polymerase tree have proofreading capabilities?
No.
Does the DNA polymerase, careful, does the DNA polymerase have proofreading?
Yes.
Yes, so it has what?
A 3' to 5' exonuclease.
Exonuclease activity. Excellent. So let's, if we can summarize, okay? Listen carefully, this is high yields. So we have origin of replication. The origin of replication is found by an enzyme. We call this enzyme helicase. The helicase, it breaks the hydrogen bonds, creating single-stranded DNA. The single-stranded binding proteins, they keep the strands apart. So in terms of strand synthesis, the primase synthesizes the RNA primer. So 5' to 3'. High yield. In eukaryotes, DNA polymerase delta and alpha, they extend the primer and they have proofreading abilities. They have 3' exonuclease activities. You're going to learn in term 3, in terms of microbiology, that DNA polymerase 3, it also extends the primer. What's the difference between the leading strand and the lagging strand? In the leading strand, the helicase leads the polymerase, 1' 1 polymerase. In the lagging strand, the helicase and polymerase are opposite, and you have fragmented replication of DNA. You have multiple primers there, you have multiple polymerases there, thereby inducing more errors. How am I going to remove the primers? You remove the primers by RNase H, which has a 5' exonuclease, which digests the primer. These are in eukaryotes now. The DNA polymerases, they replace the primer with DNA. In the lagging strand, the DNA ligase, it joins the fragments. This is important for lagging. And for micro tie-in, the DNA polymerase 1 has both removed the primer, so it has a 5' exonuclease activity, and replaces RNA with DNA. So in terms of the direction of your replication fork, you have the topoisomerases. So it unwinds the DNA via negative supercoiling to relax and prevent breaks. You have fluoroquinolones. These are antibiotics, so double-stranded DNA fragmentation. It inhibits DNA gyrase or topoisomerase 4. These are subtypes of your topoisomerase 2. You can also have topoisomerase 2 inhibitors, your posides, your etoposide, your tinoposide. These are anti-neoplastic agents that leads to double-stranded DNA fragmentation. We're good so far? We've summarized everything. This is just an example.
Question.
Go ahead.
What's it called? Telomerase, will that be high yield on the exam?
The polymerases?
No, telomerase.
Telomerase. I think it is. What's the function of telomerase? Any volunteer?
Converts an RNA strand into a DNA strand.
No, telomerase.
Oh, telomerase?
Telomerase. At the end of DNA replication, you need to protect the end parts of the DNA, right? Because there's a chance that the end parts will going to be lost, right? So you need to add some link there, correct? So you need to add some what do you call this one? Some non-coding strand there. right? So you have telomerase enzyme. This is mutated in cancer cells. That's why you have continuous replication of your DNA in cancer cells, right? So at the end of the DNA, you need to have telomeres. The telomeres, you need to add non-coding strand. Why do we need to add a non-coding strand? So that we can protect the end of the DNA. Why? Because the end of the DNA, that's where you can have mutations, or it can be cut by the end. So you need to protect the ends of your DNA, right? So telomerase, it is a ribonucleoprotein that adds what? A repeat sequence to the 3' end of telomeres, okay? A telomere is a region of your repetitive sequences at each end of the chromosomes of most eukaryotes, right? So they add a repeat sequence to the 3' end of your telomeres. Why is this high yield? In cancer cells, you have an increased activity of your telomerase. They keep on adding repeat sequences to the 3' end of your telomeres. That's why you can have an immortal cell line. It keeps on doing replication, okay? So that's the high yield point for your telomerase, okay? When you think increased activity of telomerase, think about cancer cells. Think about you have an immortal cell line. Why? Because the telomerase keeps on adding nucleotides to the 3' end. So you have continuous DNA replication. We're good so far? Did I answer your question? Is it okay?
Yes, thank you for answering.
Yep, you're welcome. So as I've said, top one, one cut, top two cuts. Histone acetylation and deacetylation. Histone acetylation makes the DNA active. A for active. Yada, we talk about this one. This is from first aid. We talk about all of these. Know the enzyme and know the function. Okay, now we talked about DNA replication. Let's talk about transcription. Did you talk about transcription in class? Yes?
Yes.
Okay, so for transcription, right? You still have a template, you have a DNA, and we want to make what? An RNA. How are we going to do that? So first you need to have the Kaat box or Tata box. This is the start of your transcription, right? So does the enzyme that starts transcription bind to the CAAT box and Tata box? Yes or no? Does it directly bind to that box or not?
Yeah.
Careful.
No, don't you need another factor?
Excellent. So you have your initiation factors, right? So the initiation factors, they won't bind to the Kaat or cat box or the Tata box. So it's just there to make sure that your RNA polymerase knows that after that cat box and Tata box, that's the start of the transcription. So the RNA polymerase doesn't actually bind to the cat box and Tata box. They're just, think about this as a signal sign, right? So you start here. How do I know that I'm going to start there? You need to look for a signal, right? The RNA polymerase needs to look for a signal. I see the cat box and the Tata box. So I'm gonna bind after the cat box and Tata box. Okay? So don't forget, they don't bind exactly to the cat and Tata box. They bind after that. So the transcription starts after your cat box and Tata box. And then the RNA polymerase, you add complementary RNA nucleotides to the template DNA strand. The DNA strand is read in what direction? Again, high yield.
3' to 5'.
Excellent. And the RNA is synthesized in what direction?
5 to 3.
Excellent. 5 prime to 3 prime. And then, what's the difference in DNA replication and RNA transcription?
RNA doesn't need a primer.
Remember the nucleotides in the DNA you have thymine, right? Thymidine. Here you have uracil, correct? For your RNA. So let's summarize. Okay. So the RNA polymerase, you're looking for a signal. Found the cut box and data box. I'm gonna bind after that one and start your transcription. The RNA polymerase, it adds complementary nucleotides to your template DNA strand. The RNA strand is identical to the other coding DNA strand except high yield uridine. The U is substituted for thymidine, right? The nucleotides are only added to the 3' end of your RNA molecule. And then the various proteins, they bind to a sequence. For example, AAU, AAA, or here, the polyadenylation signal, AAT, AAA. You're the 3' end of the pre-mRNA molecule, about probably, let's just say, 30 nucleotides, right? Downstream, the cleavage and polyadenylation signal, which cuts the pre-mRNA. And then it will gonna become, you need to splice the pre-mRNA, a cap, a 5' cap, and a tail are added to the 5' end and the 3' end of the new RNA molecule. Why? For protection and to help it leave the nucleus. So you need to have the 5' cap and the poly A tail for this mRNA to leave the nucleus. The 5' cap consists of single G nucleotides, and the 3' tail, the poly A tail, consists of hundreds of A, your adenine nucleotides. Okay, so the 5' cap makes it so both ends of the mRNA are protected from exonuclease. That's why you have a 5' cap. Function of it to protect the mRNA from exonuclease, which targets the 5' end. The non-coding introns, right? They are removed from the RNA strand, and the remaining coding sequences, your exons, are joined together, right? And the 5' cap, another high yield point, they can be recognized by the nuclear pore complex, which allows the mRNA to leave the nucleus. So that's your transcription. So you have transcription there. When is the start? After ka tata box, RNA polymerase, blah, 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 blah. You have synthesis of your mRNA. It has both exons and introns there. Then you have the stop there. This pre-mRNA, it needs to be spliced. Why? Because it contains both introns and exons. How am I going to remove the introns? Anyone? How am I going to remove the introns?
Spliceosome.
Spliceosomes, exactly. So you form a lariat structure. GU, AG, you form a lariat structure to remove the introns via your spliceosomes. Then, after the introns are removed, is that it? No. You need to have a 5' cap, and the poly A tail. Why do I need a 5' cap? So that my mature mRNA can't be digested by my exonucleases. The 5' cap is also a signal for your ribosomes to come in and initiate your translation. What if I don't have a 5' cap, and what if I don't have a poly A tail? Can the mature mRNA exit the nucleus via the nuclear pore? No. You always need to have both the 5' cap and the poly A tail to exit the nucleus via your nuclear pore. Okay? I have a question. How are we going to regulate transcription? how am I going to regulate this transcription? It's on top of this slide. Look, I have an enhancer and a silencer. What's the function of an enhancer and a silencer? And think about it conceptually. Where can I usually find these enhancers and silencers? So give me the definition. What is an enhancer? What is a silencer? And where can it usually be found?
Enhancers are usually upstream, and they usually help recruit other mechanisms to help you boost that transcription rate.
Exactly. So it increases the rate of your transcription, your enhancer. Silencer, it decreases the rate. Actually, it can be found upstream, right? You're right, enhancer. And silencer, usually downstream. So think about it. You're in the nucleus now, right? And you're floating around. These enhancers and silencers, you're in a liquid environment. They are near the promoter, and some of them are far away from the promoter. So think physiologically, how am I going to interact with the promoter region as an enhancer and silencer? For example, I'm far away from the promoter.
You form
how
a loop
am I going to enhance? How am I going? Go ahead.
So you form a loop structure, the DNA loops around, and you basically have the protein that binds the enhancer binding to that initiator complex to increase transcription.
Excellent, excellent, very good. Remember, you're in a liquid environment, right? In the nucleus, so it loops around in order to go to the promoter. Excellent. Let's see. So high yield again, RNA processing, 5' cap and polyadenylation, 3' end, and you need to splice out the introns. The exons exit the nucleus. Exons exit the nucleus, and the introns are retained inside the nucleus. Voila, how am I going to remove the introns? Spliceosomes. You have GU and AG, which forms a loop intermediate. We call this one the lariat structure, right? If you remove the lariat structure, then you have a phosphate bond between the two exons. Okay, we're good so far? We're cool, okay? Do you need a break? Or let's move on.
Move on.
Move on, okay.
Move on.
I like that attitude. So protein synthesis now. So we have mRNA. We remove the introns. We are now exiting the nucleus. We are now in the cytoplasm. So here you have protein synthesis. So how am I going to make, I think I'm missing something here. Did I miss something for protein synthesis? Maybe at a later time. So the difference between eukaryotes and prokaryotes. The difference in the ribosomes. So eukaryotes 80S, even, right? Prokaryotes 70S, right? They're prime numbers. S just means Svedberg units, right? So when you get the ribosomes and you measure them by a centrifuge, so you have Svedberg units. The amount of precipitation is what we call the Svedberg unit. So it's different. Eukaryotes 80S, right? 80. Prokaryotes 70 S. So initiation. You have the initiation factors. They identify the 5' cap. That's why the 5' cap is very important. And then your initiation factors, they assemble the 40S with the initiator tRNA. Let's talk about the tRNA. Yes, that's the one that I'm missing. So the tRNA, right? So you have 5' and 3' end. How do I know that the vignette is describing a tRNA? How do I know that a vignette is describing a structure of your tRNA?
You know that tRNA does not have N-formal methionine.
Excellent, excellent. Don't forget, there are different kinds of your cytoplasmic RNA. Okay, tRNA. This is high yield. Your transfer RNA. This is the most abundant, but the smallest. I know, guys. So T for tiny. tRNA, transfer RNA, think most abundant but smallest. T for tiny. Your rRNA, your ribosomal RNA, this is the most abundant, right? Think robust rRNA. mRNA, this one, it carries protein code to the cytoplasm. They are the largest RNA molecule. M for massive, okay? Those are high yield types of cytoplasmic RNA. tRNA, tiny, second most abundant. rRNA, robust, they are the most abundant. mRNA, massive, they carry protein code to the cytoplasm. Then, where does the protein attaches to the tRNA? At what end do you think the proteins attach to the tRNA? Is it at the 5' end or at the 3' end?
3'.
3' end. And could you give me the codon, the triplet codon for that? The 3' end of your tRNA.
AUG.
Where the protein will gonna attach to the 3' end. If the protein sees that one on the tRNA, it attaches there.
CCA.
Excellent, very good. CCA, think can carry amino acids. So on the tRNA, the protein, it attaches to the 3' end. It looks at the triplet codon, the CCA. Your can carry amino acids. So you have that. You have the initiation factors. It assembles the ribosomal subunits. All right, this requires GTP. Now let's look at the image here. Lower image, right? So you have GTP for tRNA gripping and going places. That's high yield for translocation, and ATP for charging. If there is something wrong with ATP, with charging, right, of your tRNA, there is a mistake in the carrying of a protein, okay? If there is wrong with ATP, there must be something wrong with activation of your tRNA or the charging of the tRNA, meaning there is a mistake in the binding of your protein to your CCA at the 3' end, okay? That's ATP, charging. There's something wrong. Now let's look at the image here on the lower right side of the slide. So we have 3 sites, E site, P site, and A site. Remember, the ribosomes here, they're going to the right, okay? So as always here, you have the 5' to 3' codon. It's being read by your ribosomes in 5' to 3'. You have the anticodon in your tRNA. The tRNA is carrying the appropriate amino acid in the 3' end. You have the CCA there, right? You have 3 sites, A site, P site, and the E site. What's the difference? The A site, this is where you have the incoming amino acyl tRNA. The P site is where you grow the peptide. The E site is for empty tRNA. High yield question. The first tRNA carries what protein? The first tRNA carries what protein in eukaryotes?
Methionine.
Right? Methionine. In prokaryotes?
FMET.
FMET, 4-mil methionine. High yield question. Where does it go, the methionine? The tRNA, the first tRNA that carries methionine, where does it go? A site or E site?
A site.
Careful.
P side.
Excellent. This is the first tRNA. So it actually goes to the P side. That's high yield point, right? It recognizes AUG for the codon. You have the anticodon UAC for your methionine. Now the second tRNA, it goes where now? Look at the site. Where do you think it goes? The second tRNA.
A site.
A site. So the second tRNA goes to the A site. And then something happens. The protein from the second tRNA, we're going to go to the P site. What do you call that enzyme that transfers the protein? Come again, please.
Peptidotransferase.
Excellent. You have the peptidyl transferase. Then this ribosome moves to the right. The first tRNA from the P site, we're going to go to the E site. The second tRNA from the A site, we're going to move to the P site. And then the third tRNA, we're going to go to the A site, and then you have the transfer again. So where does the peptide grows? On the P site. In P site, you have, it accommodates the growing peptides. How am I going to stop protein synthesis or stop translation? You have eukaryotic release factors. They release a stop codon. They recognize the stop codon. Could you give me the 3 stop codons, please? High yield.
UAA, UAG, and UGA.
Exactly. You are away, you go away, you are gone. Excellent. So that's your protein synthesis. Let's summarize for clarity, right? Different types: eukaryotes 80S, prokaryotes 70S. The synthesis of protein occurs from the N terminus to the C terminus. For the tRNA, in order to charge the tRNA, in order for the bond between the protein to attach to the CCA at the 3' end of your tRNA, you need ATP. For the translocation of your tRNA, you need GTP. So 3 phases of synthesis: initiation, elongation, termination. Initiation, you have the eukaryotic initiation factors. They identify the 5' cap. They assemble the ribosomal subunit. This requires GTP. Then you have the elongation. The first tRNA, which carries methionine, goes to the P site. The second goes to the A site. Transfer of protein from the A site to the P site via peptidyl transfer. Race, the ribosomes advances 3 nucleotides towards the 3' end of your mRNA, moving the peptidyl tRNA to the P sites. How does it know to stop synthesis? You have eukaryotic release factors recognize the stop codon. You are away, you go away, you are gone. And halt translation. The completed polypeptide is released from the ribosome. This requires GTP. And where do you think it goes? Is that it? Is that the end of the protein? Where does it go? Where do you think it goes? Anyone?
It depends on whether or not that mRNA has a special site for it to go to the rough ER, or if it's just in the cytoplasm.
Excellent. So the protein, right? Let's clarify the. So you have the protein. It can either go to your Golgi to modify the protein. What if I have a defective protein? I made a mistake. I don't want this protein anymore. What would I do?
You tag it with a ubiquitin, and then you degrade it.
Excellent, excellent, excellent. Very good. So ubiquitination. You tagged it with ubiquitin, and that will gonna go to where? What organelle?
Proteasome.
Proteasomes, exactly. So that it can be degraded. Excellent. So A site, P site, E site, you know that already. We're good so far?
Yep.
Yep. You're good? Okay, let's do this one. Mitosis, right? Okay, basic biology from high school, right? Interphase, prophase, metaphase, anaphase, telophase. Okay, tell me, prophase, what's happening there? They're going to describe you in the vignette. So how do I know that we're in the prophase?
Nuclear envelope.
Nuclear envelope comes apart.
What else? You have a condensation of your chromosomes. What else? Formation of your mitotic spindles, right? How about metaphase?
See them line up in the middle of the cell with their centrosomes on their side.
Exactly. So you have your kinetochores there, you have your centrosomes there, right? Don't forget colchicine can be added to this phase to arrest the cell in metaphase for cardiotyping. After this, you have anaphase. Anaphase is when sister chromatids separate. And then you have the telophase, your contractile ring, where the nuclear envelope reforms as the chromosomes condense and the nucleolus reappears. And then you have cytokinesis, your cleavage furrow. These are your actin filaments here. The spindle disassembles, and then you have the daughter cells, or a diploid cell. Contrast that one with meiosis. In meiosis, where can you find mitosis, by the way? What cells undergo mitosis?
Somatic cells.
Somatic cells.
Nearly every cell in the body, except
germ cells.
Except germ cells, right? Your gametes, your oocytes, and your spermatocytes, right? So meiosis 1, it's the same thing as mitosis. Think about meiosis as doing mitosis twice, right? So you have still the same phases, PMAT, that's the mnemonic, prophase, metaphase, anaphase, telophase. You do it twice, right? So first, you form a tetrad. You have a paternal and maternal homolog, right? Each replicated chromosomes, they pair with its own homolog. The tetrads, they line up at the metaphase plate. And then you have crossing over, right? Exchange of genetic materials from the paternal and maternal homologs. And then you separate them in anaphase. And then you have 2 haploid cells at the end of meiosis 1. What's high yield here is the eggs, the oocytes. They are arrested in prophase 1 of meiosis 1 until puberty. Okay? So at the end of meiosis 1, what happens? Each pair of homologous chromosomes is split. Their tetrads are now split. By meiosis 1, you have reformation of your nuclear envelope, and the chromosomes condense at the end of meiosis 1. Is that the end of meiosis? No. You need to go another round, right? For meiosis 2, it's the same process. You have PMAT, but it's meiosis 2. Next, there is no further DNA replication before meiosis 2. Prophase of meiosis 2 is just like a mitotic prophase. And then the chromosomes line up at the metaphase plate. Second cell division follows, and it results in your 4 haploid daughter cells. Meiosis 2 is similar to mitosis in that sister chromatids are segregated into separate daughter cells. So you start with homologs, right? And then you end with haploid cells. Contrast this one with mitosis, wherein you start with diploid and you end with a diploid cell. Okay? So this is another good picture. Compare and contrast mitosis from meiosis. Where can you find crossing over? Or synapses? Where can you find that? Only in
prophase.
Only in meiosis. Where can you find tetrads? If in the vignette it says tetrads, it is only in meiosis, right? Where can you find haploid cells? Haploid cells only in meiosis. Okay, make sure you know the contrast between mitosis and your meiosis. PMAT, don't forget, same thing. And meiosis, you do it twice, reserved only for your gametes, your oocytes, and your spermatocytes. All right, let's do some physiology. You're all good? I know it's 12 o'clock here in the East Coast. You're all good?
We're good.
We're good. Do you want to go? Do you want to do physiology?
Yeah.
Or do you want to take a break?
Let's do it.
Keep going.
Okay, let's go. So, Conductance, resting memory potential. Give me the voltage. What's the voltage for the resting memory potential?
-60.
Careful.
Oh, -50. No.
Careful.
-70.
70. Careful. Okay, guys. I know this is not a trick question, right? So -70, right? So in order for action potential, what do I need to do? That -70, it needs to be less negative, right? It needs to be positive. Excellent. And how am I going to do that? By opening up channels in the cell. Question, what's the major intracellular ion in the cell?
Potassium.
Potassium.
What's the major extracellular ion in the cell?
Sodium.
Sodium.
Sodium. So where does, if I open my sodium channel, where does it go?
Into the cell.
Into the cell, right? 3 sodiums in, 2 potassiums out. So action potential. First, look at the image here. So we have numbers. 1, 2, 1, 2, 3, 4, 5, 6, 7. 1, sodium channels, they start to open, right? Meaning the positive charge, it flows into the cell. So it causes a slight depolarization. So -70, it becomes less negative. Now, a threshold is reached. This is where all the voltage-gated sodium channels, they begin to open fully, right? So meaning more sodium channels are going into the cell and further depolarization. So look at 3, 3. Look at this. You have rapid depolarization. This is due to sodium conductance. Due to sodium conductance. Question, where can I find the point of highest sodium conductance?
4.
Peak.
Excellent, at the peak, right? And then what happens after I fully opened my sodium channels? All of the sodium are now saturated inside the cell, right? What happens next?
Your voltage-gated potassium channels have a slower opening time. So by the time you reach 4 or 5, like that's when all the potassium channels start opening, and that's when you start getting your repolarization.
Exactly. So you are delaying the opening of your potassium channels. You are closing the sodium channels, but remember, your potassium channels, sometimes they open delayed, right? So you open the K plus channels, what happens? K channels rush back into the cell. Why? What's inside the cell? The charge inside the cell is now more positive, right? You want it to go back to negative. So look at 5. So the channel, the sodium channels, they begin to inactivate, and the sodium conductance falls at the same time as the K plus conductance starts to develop. Now, look at Sais. Look at number 6 there. It seems like it goes way, way more negative. Instead of going to -70, it shoots to -80 or -90, lower than -70. Why do you think that happens? It looks like it overshoots. Look at Sais. 6.
Yeah, because those voltage-gated potassium channels open and close more slowly, by the time you reach your resting memory potential, those potassium channels are still open. That's why you get that hyperpolarization.
Excellent. Because of that, very good, very good. Because of that delay in your K plus channels, in closing, it sometimes overshoots, right? Where is the point of the highest potassium conductance?
At 6.
Where?
Maximum potassium conductance is at 5. Like,
excellent, at the end of your 5, right? Because that's where you are overshooting now. So the point of highest sodium conductance is 4, and then the point of your K conductance is actually at 5, where the channels are opening. Now I have a question, guys. Look at the image. So there's some underneath this action potential, right? There's an area below 2 and 5. What do we call that period? Between 2, between 2 and 5, you have an area. in between them. What do we call that period?
Absolute refractory period.
Excellent. Absolute refractory period. What do you mean? If the cell is in absolute refractory period, what does it mean?
It means that you can't generate another action potential because the gate channels, I kind of say, are tired or are blocked.
Exactly. So you cannot have another action potential. Is that beneficial to the cell? For example, the neurons, is this beneficial? Yes, of course. Or else we're going to have epilepsy, right? You're going to have tonic-clonic contractions every time. Okay, this is good for the cell. Now, question. The graph, an area below 5 and 6 up to 7. What do we call that? You have your relative refractory period. What is the significance of that relative refractory period? During hyperpolarization, during SAIS, right? During 6, hyperpolarization of the cell, you have a relative refractory period. What's the significance of that relative refractory period?
It means that you can possibly generate an action potential, but because there's more potassium coming back into the cell, it might be difficult.
Excellent. Very good. So the point there is you can actually have another action potential. Now let's do some, let me quiz you. How am I going to initiate another action potential in the relative refractory period? What needs to be done to the stimulus?
It has to be a higher or stronger stimulus.
Excellent. You either increase the strength, right? Increase the amplitude of your stimulus. What else? Continuous. You need it to be stronger and continuous, pulsatile of your stimulus, okay? That's very, very good. Know this one. This is high yield. And then 7, the channels reset to the normal conditions. So let's summarize, right? If I have an action potential, what happens? Sodium channel opens. Sodium rushes into the cell. Potassium goes out of the cell, until the point that the inside of the cell is saturated with sodium. Now I need to close the sodium channels and start opening your potassium. Remember, they open slowly and close slowly. The potassium rushes into the cell and it overshoots, right? It becomes less, it becomes more negative. Why? Because it's delayed. Your K channels closing are delayed. It overshoots, but then it resets back to normal. Don't forget, absolute refractory period versus relative refractory period. Absolute, you cannot have action potential there, but in a relative refractory period, you can have an action potential just by increasing the stimulus and the amount of times you're going to initiate the action potential. Good job. High yield, difference between hyperkalemia and hypokalemia, put a star here. In hyperkalemia, you have a depolarized state, meaning the K conductance increases, right? However, there is no resetting of sodium channels, right? The memory potential approaches 0 and there results in flaccid paralysis. Contrast this one with hypokalemia. It causes a hyperpolarized state. The threshold is never reached. The memory potential approaches K potential, results in flaccid paralysis. What's the high yield point? Both of them has flaccid paralysis, right? But what is the difference? In hyperkalemia, there is no resetting of your sodium channels. Why? You have a slow depolarization. That's in hyperkalemia. And the membrane potential approaches 0. In hypokalemia, the threshold is never reached. The membrane potential approaches the K potential. Okay, that's hyperkalemia versus hypokalemia. And I have some questions. I think that's the end of it. You can go over the MCQs. Thank you for attending today's session. Do you want me to go over genetics?
Yes.
Yeah, we can go over.
That'll be cool, yeah.
Yes.
Yep. Can we have a break for 10 minutes? I need just to drink some water.
That's fine, thank you.
Thank
All
you.
right. See you in 10 minutes.